Hello students, now I am talking about the last part of the receptor concept, that is receptor concept part 3, and in this I will discuss remaining two type of receptors. So let's begin with the third receptor is the enzymatic receptors, right? As you have seen in the previous receptor that they the receptor with intrinsic ion channel, that receptor was having a receptor inbuilt with the ion channel in it, right? So ion channel was with the receptor, right, in the same structure. Now in the third type of receptor, enzymatic receptors, the enzyme is in the receptor itself. Okay, as you see in the GPCR, the receptor and the ion channel, or the receptor and the enzymes, are two different places, at two different places. That's why the GPCR needs the alpha units to go and get activate the enzyme and the ion channels. But in the second type, the channel with the intrinsic ion channel, the ion channel was within the receptor, right? So that's why there was no need of the second messengers to go and do that. Now in the third receptor, even the enzyme is also in the receptor itself, so no need to go and activate the enzyme in somewhere else, right? The enzyme is inbuilt with the receptor itself. So these receptors called as the enzymatic receptors. They are also called as the ligand regulated transmembrane receptors with enzymatic cytosolic domain. They also been called as this. So or they call it as intrinsic enzyme receptors, right? Sometimes call them as transmembrane enzyme linked receptors. Okay. These, if you see the one diagram here, one part, upper part, you can see the ligand, and the down part means this whole receptor one. It's a whole receptor. If you see, this is a whole receptor, but the upper part of the receptor is having the ligand binding site, and the below part, which is a intracellular part or inside the membrane part, that is called the uh, transmembrane domain. So this is a kind of transmembrane domain, or below the membrane. This whole part is the having the enzymatic mechanism or enzymatic properties. Okay, now we'll see further, you will understand well. These receptors are having various variants, right? Because the different type of receptor, different type of enzymes are attached with them, right? Differently they attach with them. That's why on this basis they are having various type of. So here you can see they can be grouped into the different 5 type of receptors. First one is the receptor tyrosine kinase. In this, the enzyme which is attached with the receptor is a tyrosine kinase enzyme. For example, insulin receptors, BCR, ABL proteins receptors, EGFR and VEGFR receptors, they all receptors are of this type. They are all having tyrosine kinase as a intrinsic enzyme. In this also there are two categories are there. One we call it as receptor with intrinsic tyrosine kinase, and another is the receptor with intracellular protein tyrosine kinase, JAK-STAT. Means in this one other protein of tyrosine kinase is going to be attached, that is the JAK and STAT. These are two different proteins. JAK is a one protein, STAT is another protein. So they are going to attach with it. So in this way they are of two types. Second type of it is the receptor tyrosine phosphatases. So there are, in place of tyrosine kinases, they are tyrosine phosphatases, right? They used in the immune receptors as CD4 on B and T lymphocytes. Third type of receptor is called as the non-receptor tyrosine kinase, right? Here also SRC, genus kinase, ABL type are using this kind of receptors. Fourth one is called the receptor serine threonine kinases, right? So here serine threonine kinase enzyme has been attached with the receptor, for example TGF beta. Receptor guanylyl cyclase, this is the fifth type of, here the enzyme which is attached with the receptor is guanylyl cyclase, right? Example B type natriuretic peptide, BANP. So these are all 5 different type of these things. In this first category we will discuss in this video, that is the receptor tyrosine kinase, right? Receptor with intrinsic tyrosine kinase activity or intrinsic enzyme receptors. Here we have to understand how they look like and what is their functions. So this class of receptors have a subunit with enzymatic property or bind a JAK genus kinase enzyme on activation. So as I told you, both type of things are going to come in this. Either the receptor having the enzymatic property or they bind with the JAK and genus kinase. The spelling of genus is J, comes at here, it's written G. So genus kinase. The agonist binding site and the catalytic site lie respectively on the outer and inner face of the plasma membrane. Means agonist binding site is present on the outer surface and the catalytic site is on the inner surface. Okay, because enzyme is present in the inner surface, that's why it is playing a role in the catalyzing the reactions. There are 2 major subgroups of such receptors, as we have already told you. Those that have intrinsic enzyme activity and another those which are activating and bind with the JAK and STAT kinases. Now you look into this structure, this diagram here, and this diagram I have taken from the Rang and Dale. You can see in this structure, you see this structure, right? So it has distinct 3 parts in it, right? One, this particular part, another this particular part, and then this particular part, like that, 3 distinct parts. The first part, which has been shown by blue color here, that is a receptor domain. So this is part we call it as a receptor because this is the part which is going to bind with the agonist. The middle part is a transmembrane alpha helix, which is attaching the third part with the first part, right? So transmembrane alpha helix, it is transmembrane, it is present in the entire membrane. Tyrosine kinase domain, this is the third one, this is the enzymatic domain, right? Actually this is a receptor, this is an enzyme, that's why this whole unit we call it as an enzyme, a receptor with intrinsic enzyme activity, right? So it is having a tyrosine kinase domain. So like that these receptors will look like. Usually what happens whenever the growth factor, for example here the agonist has been shown as a growth factor, right? It can be anything, here it's shown as a growth factor. So one agonist is binding on the receptor domain. Once they bind on the receptor domain, the receptor unit get activated and it will go for the dimerization. You can see it will go for the dimerization, means the one unit will make its own one more monomer. So one monomer makes one more monomer, it means it's become dimer. So now they will dimerize and make a double unit of it. Once it is happening, now next you need to activate the enzyme. So once the enzyme get activates, then it will do the further action. So how the enzyme will activate? This enzyme will activate by itself. Once the agonist binds with it, dimerization completes, then what happened? This enzyme phosphorylates its own, sometime like this, sometime like this, or sometime by their own, right? So likewise, the enzymatic domain, the tyrosine kinase, will phosphorylate itself. Phosphorylate means one phosphate comes and attach with it, because phosphate is an energy. So when phosphate attach with this enzyme will get activated, right? So now once this autophosphorylation happens, the enzyme has got the activation, right? Now this activation, what the enzyme has got the activation, now it will attract certain proteins, right? And this attraction of certain proteins will initiate one uh, cascade of events. For example, here it has activated one protein which we call it as GRB2, right? Activated a GRB2. So now GRB2 get activated, and this GRB2 has activated another protein which we call SOS protein, right? SOS protein. Now this SOS protein is also got activated, and now this SOS is activating GTP, or sometime this GRB2 will activate the RAS protein, and the RAS will activate this GTP to GDP conversion, and the activation begin, right? So either the activation begin by the SOS or by the RAS. After this activation, what will happen? The RAF protein will get activated. In the previous receptor also I have told you about this. So RAF protein get activates. This RAF get phosphorylation, then it will activate the MEC protein. MEC will activate the another MAP kinases, and like that the phosphorylation begin. Like, if you see here, it's a cascade of events. They all are the kinases. So RAF has activated, then MEC activated, then another MAP kinase activated, and finally it reaches to the gene transcription, right? It reaches to the nucleus, and there it changes the gene transcription process, and then it will change the protein synthesis. Likewise, the different proteins will be formed. So what you have seen, the agonist which has bind here, agonist which binds here, will ultimately reach to the effect of that agonist, right? The response of that agonist by these consecutive processes, it will reach to the DNA, and it will change the gene transcription and will change the protein synthesis, and the different kind of proteins will form. Okay. So I hope you understood that how it's all happening. So here in this process, the phosphorylation is a very important part, because the tyrosine residue or the enzymatic domain, if it is not get phosphorylated, it will not begin the further process. It will not attract or activate the further proteins. So first the phosphorylation has activated the enzymatic domain, and because of this activation, GRB2 protein has attracted towards it, and the GRB2 protein get phosphorylated, then it will activate either RAS, or through SOS it will activate the process of activate the further proteins like RAF, MEC, MAP kinase, and here also I tell you, here also they are not only, it is shown you like RAF, MEC, MAP kinase, there are so many other proteins are activating in between, right? So after, because it's a very controlled and very systematic process, very complex process, and after that it reaches to the nucleus, because the nucleus contains the DNA. Anything which is going to affect your DNA will change something seriously in your body. That's why the body has a one very proper mechanism. Without going in a sequential activation of certain proteins, no any agent can reach to the DNA, because once it reaches to DNA, a serious changes are expected, right? That's why the hormone system of your body is having a direct regulation by this process. They use the same process to change the DNA, right? That's what the growth hormone changes your growth system, or the changes the growth in your organ system, right? That's possible only through the gene regulation. So I hope you understood how this whole process is happening. Now you see this, I have brought another diagram which can give you even clear picture of this. You can see this here. In previous diagram, you have seen the agonist was the growth hormone. Here the agonist is the interleukin 6. So now the interleukin 6, which is going to bind with the GP130. So once it binds with the GP130, here you can see this diagram explains you how these JAK proteins get activated and how the STAT protein get activated and how they perform their action. And ultimately, if you see in this part, they are changing the DNA level, right? I think this diagram will be more relevant with the next pathway, what I'm going to talk about. Now, the second type of receptors under this is JAK-STAT kinase binding receptors, right? They are also called as cytokine receptors or receptor tyrosine kinases. So what is happening here? The example is cytokines, which are going to bind with the receptor. Now you know the structure of the receptor, so you can understand well. Now once the cytokine binds on the receptor, the enzymatic domain get autophosphorylated. Once they get autophosphorylated, what is happening here? Once the autophosphorylation done, as you have seen in previous case, one GRB2 protein was attracted towards it. Now here in this case, the different protein is attracting towards it. That is a JAK protein. That is called as the Janus kinases, or sometimes we also call it as just another kinase, right? So it is one more kinase which is going to activate it. That's called this JAK, right? So this JAK protein will come and bind with this enzymatic domain, right? Once this binding of JAK completes, the JAK get phosphorylated, and this phosphorylated JAK will activate or will attract another protein which is flowing into the cytoplasm, floating into the cytoplasm somewhere. Once the JAK get phosphorylated, automatically the STAT will attract towards it. The STAT is having a SH2 domain in it. SH2 domain is present in that particular structure. And then this protein with this SH2 domain, which we call it as a STAT, is going to attract towards the JAK, right? And here it get activated. So as the STAT, which is going to attract towards the JAK, and STAT is also an abbreviation which is stand for the signal transducer and activator of transcription. STAT means signal transducer and activator of transcription. So this STAT protein is now get attracted towards JAK, and it binds with the enzymatic domain where JAK is already attached. Now this JAK, which is already phosphorylated, will phosphorylate the STAT. Now once the STAT get phosphorylated, you can see here, once the STAT get phosphorylated, the STAT will dimerize, right? The two units of the STAT will come closer and bind with each other and make the dimer, right? Now once this dimer is formed, now this STAT will activate certain other proteins in the process here, and or directly STAT can enter into the nucleus, and there it will, it will change on the gene transcription. The another diagram can also you can make it more clear. Cytokine binds it, JAK activated, JAK has attracted the STAT. Now the STAT become dimerized, here dimerized, and this dimerized STAT entered into the nucleus and changing on the responsive element of the DNA, and there it changed the gene transcription, and then it changes the protein synthesis. Okay. So in this what called the type of receptor, the difference what you have seen in before, here two different proteins are activating which were not there in previous one. Here one JAK activates, and after the activation of JAK it activates STAT, and then these STAT get dimer and act on the DNA. While in the previous receptor, you have seen that once the enzymatic domain get autophosphorylated, the phosphorylated unit of that will attract one protein called GRB2, and then further proteins get activation, and then it reach to nucleus. So this is what these two different receptors are playing the part. In this receptor, the cytokine receptor, there are we have various drugs. When we know that these pathways are very, very important, very strong pathways, they can change the DNA, they can change the protein synthesis, right? They can change the whole course of physiological reactions. So they are involved in various kind of diseases as well. Once they get malfunctioned, various diseases happens. Even the cancer is the result of all these or various type of tumors are the result of all these malfunctionings, right? For that purpose, nowadays the main research is going on towards discovering various drugs which are either activating these pathways or suppressing these pathways or modifying these pathways to control their various diseases. For example, there are drugs. And now if I tell you, the JAK2 inhibitors are under development right now, and they are being developed. They're going to be developed for the treatment of polycythemia vera. Even in treatment for essential thrombocythemia, myeloid metaplasia, and myelofibrosis. On all these diseases, JAK2 inhibitors will be useful, right? I tell you some more examples here. RAS, MAP kinase pathway inhibitors are already there. For example, tipifarnib and tonafarnib. Even CRAF inhibitors are there, which we call it as serafenib. mTOR inhibitors are there, which is a very, very famous rapamycin, right? Sirolimus, and temsirolimus, and everolimus, right? So these are the drugs which are either available or under the development, and they will be used for the as a JAK2 inhibitors. Even there are some drugs, we call it EGF receptor antagonist, or we can say the tyrosine kinase inhibitor. For the previous receptor what I told you, tyrosine kinase inhibitor, tyrosine kinase enzyme. So the tyrosine kinase inhibitors are also there, where eralotinib is there, imatinib mesylate is there, dosatinib is there, nilotinib is there. Like, so BCR-ABL inhibitors are there, dual SRC-ABL inhibitors are there. So these are all the development of the drug. You all are the future researchers. If you understand these mechanisms well, in future you can develop new drugs which are acting or modulating these pathways, and then you can control various non-curable diseases. So I hope you understood what I explained you about these receptors. Now I'll move to the last receptor, which is the intracellular receptors. So these intracellular receptors, also called as transcription regulators or transcription factors, or receptor regulating gene expression. So commonly we call them as the receptor regulating gene expression, or commonly we call it as nuclear receptors, right? Even cytoplasmic receptors or intracellular receptor for lipid soluble agents. So these all are the common names of it. So we'll see further that what these receptors are. So the nuclear receptors, these are the receptors. Before this, right? The first type of receptor GPCR, second type of receptor with intrinsic ion channel, third type of receptor was the enzymatic receptors. So they all were the transmembrane receptors. You have seen they all were present the transmembrane receptor. But this is the only receptor which is not present outside of the membrane or in the membrane. They are present in the nucleus. That's why we call them as a nuclear receptors. And they are highly regulated receptors. because anything which binds with them going to change your DNA, right? So that is a one thing, that drug goes into the cytoplasm and then diffuses to the nucleus, and then it binds with the nuclear receptors, and then the whole course of change in the physiology will happen. That's why these receptors are involved in the various diseases and the disease controls. You can find it here. It is used, it means these receptors are involved in diabetes. Through that, it is involved in the cancer risk, even the obesity, free fatty acid beta oxidation, fibrosis process in the liver, in the pancreas, in the intestine, breast, prostate, inflammation process, angiogenesis, right? So tumor cell survival, and even the metastasis process, tumorigenesis. So there are various, uh, what we call it as the involvement of these nuclear receptors, very very important involvements, and they have a huge impact on the physiological system of our body, and the direct and fast changes they can cause it, right? That's why it's a highly regulated process. We'll see how these receptors are acting actually. So this is the diagram what I have taken from KD Tripathi. This will explain you that how this receptor is actually acting. I tell you here, you can see this is your hormone, right? Suppose that glucocorticoid hormone. So this glucocorticoid hormone, this is a cell, and this is a nucleus, right? This is the nucleus. So you can understand this point well. So glucocorticoid receptors, when this glucocorticoid enters inside to the cell, it binds with one steroid binding domain. You see here, this structure. This structure is having various part in it. This structure is having various part in it. If you see this particular part, so this particular part is called as the steroid binding domain. Means it is going to bind, means that steroid is going to bind on this unit. Okay, so this is called steroid binding domain. This steroid binding domain is having this carboxylic end here, right? And the other part is always the nitro end, right? So this way, this is a carboxylic end, and at the carboxylic end, the steroid binding site is available where the steroid will bind, right? In this, you can see there are three different parts clearly visible. One is this part, another is this part, and third is this part. Okay. So what does it mean? What does it mean of these three parts? So one, as I told you, is steroid binding domain. The second part is DNA binding domain, which will bind with the DNA, right? That's why this part has been given with these two hook kind of structure. We call it as a zinc fingers, right? So through this, it is going to attach or join with the DNA, right? That's why it's called DNA binding domain. And the third part is there, which is a kind of regulator, right? Which regulates the activation of this receptor. Here you can see there are three different proteins, two different proteins are also attached, which we call it as an HSP 70 and HSP 90. HSP means heat shock protein 70, heat shock protein 90. This is called IP immunophilin. So three proteins are attached here, HSP and IP. So these HSP and IP proteins are having the regulatory effects, right? So once they remove from it, the receptor is free to act on the DNA. As I told you, is a highly regulated process. Anything cannot come and bind with the receptor and changes on the DNA. That's why these controls are there. These controls will define that which things to allow and which not to allow. Okay, so these heat shock proteins, once the glucocorticoid binds with it, and receptor senses that this is the right agonist to bind with it, these heat shock proteins inhibitory control will be removed from it, and the receptor is ready to get activated. So once the receptor get activated, what will happen? Excuse me. The activated receptor will get dimerized and internalized. Here two processes are happening. One, internalization means receptor will go into the nucleus. They call internalization. After internalization, it will go dimerized, right? Dimer formation. So it will form there was a one unit, now it become two units, right? So here you can see this dimer is available. Now this dimer form of the receptor is going to attach to the DNA on a particular specific points on this DNA through these zinc fingers, right? So by using the zinc fingers, it will attach to the DNA. And why attaching on the DNA? It will modify something, and after the modification, it will form the new mRNA. Why I'm saying new mRNA? Because now once the glucocorticoid receptor activated, the receptor came and bind with the DNA, the new mRNA has been formed. This new mRNA will be having the new information, and then it will go to the ribosome and it will make the proteins by the protein synthesis process, right? And these various proteins will perform the various modified cellular functions. So likewise, these hormones are using these nuclear receptors to change the DNA and they change the protein synthesis and will form the different proteins, and these different proteins are nothing but the expression of those genes which were present in the mRNA. I hope you understood what I want to convey to you, that how these nuclear receptors look like and how these one thing I want to make it clear to all the students, these diagrams are the imaginary, right? Every author has made their own diagram, so it doesn't mean the receptor look like that, but definitely receptors are having these parts which have been mentioned here, right? So somewhat their structure look like this, but they are not exactly like this. Okay, so if you wish, you want to make some of your own diagram which is having all these similar components, you are free to do that. So these are the nuclear receptors. Now you have understood that how these nuclear receptors are acting. Now I'll tell you these nuclear receptors, you remember that in the biochemistry you might have heard somewhere the chaperones. Chaperones were nothing but the repressor proteins, right? So these heat shock proteins are the similar like chaperones, which are doing the same regulation process. Okay. Okay, now I'll move to further and I'll tell you the broader picture of it. Now if you see here in this particular diagram, you can find all type of receptors together, right? You can see receptor tyrosine kinase is here, GPCR is here, here also receptor tyrosine kinase, here integrins, they are also the one type of receptor processes, right? So they all are and as you have studied everything separately, they all are not separate, right? That's what I want to convey to you. In the cell communication pathway, nothing is separate, they all work in the same cell, and then they are having a close connection with each other, right? Here if you see, you can understand this point very clear. Here if you see, this PI3 AKT pathway, phospholipase C, G protein, here adenylyl cyclase and protein kinase activation. Same way, this is also activating the RAS, RAF, MEC, and the other MAP kinases, right? So they all are interlinked with each other, right? And that's why one is activating other is activating other, and likewise they are very much closed with each other, right? It doesn't mean that one time when the phospholipase C is activating the what called making the protein cyclic AMP, the same will be acting in some other part for the some other protein activation, right? So they are very much in working in a closed circuit, and they are very complex processes. So likewise, these all activation, finally if you see, they all activating, this is the nucleus inside, so they are activating various, you can see various proteins here. These all are different proteins. So by these different processes, different pathways, activating different proteins, and finally activating, doing the gene regulation, and this may be the result of the cell proliferation, or maybe the result of anything else, maybe the apoptosis. Okay, so likewise these processes are working in our body. In the next year you will be getting all these pathways in detail. That's why I'm taking little more time in the receptor concept, to make you understand that these are the receptors, are the basics of those cell communication pathways. Once you know the receptors, you can understand how these receptor following the path, and how these are interconnected with each other. There's another diagram which can give you the even different picture. You can see here. Here three receptors have been shown: GPCR, RTK, and integrins. You can see how much they are interrelated. I am not going to tell you all these things in detail now, otherwise you will get confused. But you can understand that once uh, these protein kinases get activates, they can activate number of different proteins, right? And these different proteins are performing various different activations, and then various different processes by different pathways. So you see, they all are interrelated each other, and these are interrelation, and these interrelations are finally reaching to the uh, what called the activation of the DNA something, right? So these are the complex processes. I'll show you one more even bigger diagram to tell you this thing more clearly. You can see insulin growth factor receptor is there, GPCR is there, enzyme linked receptor is there, right? And the ion channel receptor is there. You can see here all four type of receptors are there, right? What I told you, GPCR is there, enzyme linked receptor is there, uh, ion channel is there, and the JAK-STAT activated receptors are also there. So like that, these all receptors are playing in working in tandem, in a sync. They work with each other, and they are activating various enzymes, and these enzymes, one enzyme is activating other enzyme, other is activating other enzyme, and finally they all are having the tendency to they all are having the tendency to play acting on the DNA, and then they change the whole course of the protein synthesis, and likewise they control the whole physiological mechanisms of our body. Okay, this is another diagram, is also showing you the similar thing. I brought it various diagrams, so you can pause the video and look into clearly inside these different activated proteins and the pathways. In the next year we will definitely take in detail of all these pathways, right? Here also you can find the there are above four type of different receptors are there, and they all are interconnected with each other, and finally they are producing some physiological effect, final physiological effects. Okay, so I hope you understood this whole receptor concept. I have taken it from beginning, started from the pharmacodynamic processes, concept about the pharmacodynamic. I've given you the background, and after that slowly I started the receptors. I have explained you what are the receptors and how they act, and I've explained you the various type of receptor and transducer mechanisms, and in that you have seen the GPCR, you have seen the enzyme linked receptors, you have seen the receptor with the intrinsic enzyme activities, right, where you have seen the receptor tyrosine kinase, and you have seen the JAK-STAT activated proteins as well. and you have seen the nuclear receptors, and I've given you the broader picture where all these receptors are playing in a sync in our body, and they control each other's mechanism, and ultimate response can be produced by changing on the protein synthesis through the change in the mRNA or in the DNA. So likewise, this is a very interesting, if the molecular mechanisms of drugs are very interesting thing, because this gives you the real picture that you have taken a drug and the drug has produced its effect. Suppose that you said you have taken the insulin and the insulin has controlled your blood sugar, but how actually it happens, right? Inside, how stepwise manner it happens, that the pharmacology and the molecular mechanism gives you that picture, right? So once you understand that, then it's very easy to understand where went wrong and how the disease happens, and then you can think of on that process and that mechanism and control that mechanism which has been faulty mechanism and causing the disease, and by that you, if you develop a drug for that, there is a drug, right? So that's what the pharmacology research and the molecular research is the key factor to bring out the concept of the drug development. So I hope you all can read little more about it. There are a lot of literature available. Books are available. We have very good books in our libraries as well. Please contact me. I will supply you the books as well, and read on this, right? And please keep on posting the doubts to me. I'll be happy to answer, right? So these are the references I used for the text and the diagrams. And thank you so much for listening this whole deliberation of the receptor concept. And here we are ending with the receptor concept, right? So I hope you can revise and understand and make your concept very clear. Thank you so much for listening me carefully.